So welcome to the third presentation of Krok 1 2019 booklet, and this happens to be Histology. Histology. If you love what we are doing, support us by sharing and by liking our channel or liking this video. And of course, do well to subscribe if you are new so you don't miss anything that is being posted over here. All right, so let's begin. So histone protein synthesis is artificially blocked in a cell. What cell structure will be damaged as a result? So over here, our concern is what? Inhibition of histone production or histone synthesis. What happens when this is blocked? So it is worth knowing that histones act as, or histones are simply the chief protein component of chromatin. The chief protein component of chromatin, acting as spools around which DNA winds and playing a role in gene regulation. Now, without histones, the unwound DNA in chromosomes will be very, very long. It will be very, 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 very long. So therefore, when histone synthesis is blocked, nuclear chromatin will be damaged. And that is why over here, we are looking at C as our answer. Now, if you don't know what I'm talking about by DNA, winding, DNA, unzipping and stuff like that, please do well to, this is biology. Even it's in biology, do well to read on, what's the name of this topic itself? On chromosomes. Do well to read more on chromosomes, DNA replication and all those things. You will get the understanding much, much better. Unfortunately, I'm not doing that as a topic, so maybe another time I'll personally treat it on this channel. So if you like it, let me know in the comment section for learning purposes anyways. So over here, your answer should be what? Chromatin. Some diseases of the large intestine lead to changes in the quantitative ratio between mucosa epithelial cells. What cell types are normally predominant in cryptal epithelium of the large intestine? So we are definitely looking at what? The cells that are predominant in cryptal epithelium of the large intestine, So basically, the epithelium of the large intestine consists of crypts and intestinal villi. Crypts and intestinal villi. And they contain two types of cells. We have the goblet cells that secrete mucus. Then we have the enterocyte. So we have the goblet cells which secrete mucus, and then we have what? The enterocyte, which secrete water and electrolyte. They secrete water and electrolyte. Now, if you look at the option that we are having over here, of course, we could be looking at what? At goblet cells. So over here, your answer should be A. Goblet cells. Examination of a surgically excised adrenal gland shows large cells that can be impregnated with potassium dichromate solution. So potassium dichromate solution. Talking about the adrenal glands. So what hormone is being synthesized by these cells? Now, when the adrenal gland is fixed or is dyed or is impregnated with chrome salt. So this is actually what? A chrome salt. That's the potassium dichromate solution. It's a chrome salt. When an adrenal gland is fixed or is being impregnated with this thing or with the chrome salt, basically it allows, or inside the adrenal gland, we have what we call stored catecholamines granules that are there inside the medulla, to be specific, inside the medulla of the adrenal gland. We have the catecholamines that are being located, and these catecholamines would then be oxidized to a brown color. So when you put in the potassium dichromate solution or the chrome salt, it turns or it oxidizes the catecholamines to a brown color. And that is why sometimes we call these cells as the chromaffin cells. Chromaffin cells, coming from the word chrome salt. Chromaffin cells. So that's another name that we will give to the catecholamines or the medulla is what? The chromaffins. All right. So, and that is also over here, definitely you should talk about the function of the catecholamines. So what would they be produced? Of course, they'll be producing the adrenaline and then the non-adrenaline, or the epinephrine and the non-epinephrine's, isn't it? So that is over here. Your answer should be the adrenaline. So B should be your answer. An inflammation can be characterized by hemocapillary dilation in the affected area, decreased blood circulation, and increased vessel wall permeability. What cells play a role in this process? What cell play the role in this process? So what they are talking about is what cells are simply responsible for dilatation of the vessels, decrease blood circulation, and increase the vessel permeability. That is all. They want to know what cells are responsible for these things. But of course, before you can talk about the cells, you can even talk about the hormone. And what hormone comes to your mind? Of course, you could be looking at for what? For histamine. Histamine, isn't it? You could be looking at for histamine, because histamine is a vasodilator. And it also increases the vessel wall permeability. It increases vessel wall permeability. So over here, you could be looking at a hormone called what? The histamine, or a substance called the histamine. And histamine can be, I know histamine, most of you know that histamine can be coming from the mast cell. They can also be coming from the basophils. And usually in allergic reactions, you will see these things present. In allergic reactions, you are going to see these things present. So over here, we could be looking at a case of what? Tissue basophils, because these basophils contains a vasodilator called the histamine, which of course is what they are looking for in this question. So over here, your answer should be what? Should be E. A histological specimen demonstrates a vessel with the wall that consists of endothelium, the basement membrane, and a loose connective tissue. This vessel belongs to the following type. We've already discussed about all these things. We've discussed about the types of vessels. Of course, we have the artery, we have the veins, isn't it? Then we have the muscular type, and then the elastic type, then the non-muscular type. All of these things, we discuss them thoroughly in one of our bases. In one of the bases, of course, histology basis, we discuss it extensively. So please do well to watch those videos. They have been uploaded. So do well to watch these videos. And of course, it's worth adding that all these questions can actually be found on the Medent website, which I'll put a link in the description. So at the end of the day, you can go in there, check them out, because every question comes with explanation. For example, a question like this, the explanation is down there for you to see what it's all about. And it helps in understanding. So please do well to get hold of it. And again, on that website too, you will also see pre-Kroks, actually time-bound pre-Kroks, whereby you don't just answer, you answer but then you are restricted with time. 1 hour 30 minutes for 100 questions. I think it's fair. All right, that was just by the way. So quickly, let's brush our mind. I mean, let's refresh the memory a little bit. So basically, we have 3 layers. So we have the so for example, assuming this is a vessel, okay, this is a vessel, okay. These are walls. I'm just drawing the walls for you. Okay, so inside here, inside this vessel or inside the lumen, we have the tunica intima. That's the inside tunica intima. And under the tunica intima, we have the endothelium, we have the basement membrane, we have the loose connective tissue or elastic tissue. And the reason why I brought that there is because it helps you to differentiate between whether it's a vein or it's an artery. Then we have the tunica media, that is the middle layer. It either consists of muscles or it consists of elastic tissue. In arteries, actually, in arteries, you will either see muscles or you see elastic tissue. So if it is made up of more of elastic, we call it what? An elastic artery. If it is more of muscles, we call it what? The muscular artery. And sometimes too, we have very few muscles. So in that case, we could be talking about what? The non-muscular vein, because every artery has either elastic or muscle. So in case of very few muscles, then we could be dealing with what? With a vein. Then we have the external part, or the tunica adventitia. Tunica adventitia, or the outer part. Now, in a vein, it is made up of a lot of connective tissues, a lot of what? Connective tissue. Is very thick in vein. Very what? Thick in vein. And it is the opposite when it comes to arteries. Okay, so in arteries, the adventitia will be a little bit thin or very small, whilst the media or the middle part will be very what? Big. So these are the differences between them. And again, like I said, when it comes to the how do you call it? The intima or the inner layer, it is lined by endothelium, basement membrane, and loose connective tissue if it is a vein. Okay, loose connective tissue if it is a vein. So please take note. And then if it is an artery, it will be made of what? Elastic tissue, a bit of what? Elastic membrane, will be there. So again, we have established that this is definitely not an artery, but what? A vein. And since there's no mention of muscles there whatsoever, then we could be looking at the case of what? Case of a non-muscular vein. So that's over here. Your answer should be E. Parenchyma of an organ is composed of a pseudo unipolar neurons localized under the capsule of connective tissue. The central place belongs to nerve fibers. Name this body. The central part. The central part belongs to the nerve fibers. Name this body. So over here, we are talking about a pseudo unipolar neurons. So usually the cell body of a pseudo unipolar neuron is located within the dorsal root ganglion. The dorsal root ganglion, also known as the spinal ganglion. That is where the neuron is located. Now, the axon leaves the cell body, that is from the dorsal root ganglion, into the dorsal root, where it now splits into 2 branches. It splits into 2 branches. The central branch will go to the dorsal column of the spinal column of the spinal cord, where it synapses with other neurons, whilst the peripheral branch will travel through the distal dorsal root into the spinal nerve all the way to the skin, the joint, and the muscles. So please, this is very, 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 very important for you to understand. That's what they call it, what pseudo unipolar. It's as if it's just a one axon. But no, the axon will start dividing, or it divides into two parts, the central part and the peripheral part. And the central part will enter into the dorsal column of the spinal cord, whilst the peripheral part will also go to the distal, that is away from the dorsal root. Away from the dorsal root. So over here, they are talking about the central part, or the central place belongs to the nerve fibers. Name this body. Name this body. So over here, we could be looking at a case of what? The spinal ganglion, because that is where the cell body is located. That's where the cell body is located. So please take note of that. So over here, our answer should be A. A historical specimen shows significant amount of mucous connective tissue, vessels, as well as residual yolk and allantois. Name this organ. Name this organ. So basically, the clue here that you have to know is the fact that there's what? A mucous connective tissue, also known as the Watkins jelly. The Watkins jelly. And as I tell you in a moment, why they call this the Watkins jelly, or why our answer should be what we are thinking about. But basically, this jelly is, how do I put it? It's like a covering. Okay, it's like a covering that covers the umbilical cord. It covers the umbilical cord. In other words, the umbilical cord is buried within this jelly. It is buried within this jelly. Okay, and of course, we know the umbilical cord is more of a conduit between the developing embryo or the fetus and, of course, the placenta, which connects to the mother. And it contains two, some of you don't know this one. The umbilical cord contains two arteries and one vein. Two arteries and one vein. Like I said, all these cord is buried inside this jelly. Inside this jelly. And of course, again, it contains some remnant or some residuals of the yolk sac and then what? The allantois. So please, that is why over here we are looking at a case of what? Or we're looking at an umbilical cord to be our organ of importance in this question. So over here, your answer should be A. All right, so basically this is the end of histology. Very, very, very short video, isn't it? So please keep watching because I'll be uploading the next video shortly. All right.